The overall goal of this Western blotting protocol is to perform Western blots on small numbers of cells.
This method can help answer key questions in the hematopoietic stem cell field, such as the presence or abundance of specific proteins or activation of signaling pathways. The main advantage of this technique is that it requires fewer than 20,000 cells. Thus, fewer animals are needed to analyze rare populations of primary cells. We first had the idea for this method when we were trying to find ways to reduce the number of mice we needed to use for hematopoietic stem cell analyses. Demonstrating the procedure will be Xiongwei Tsai, a postdoc from my laboratory.
Use a swinging bucket rotor to centrifuge 1.5 milliliter tubes containing sorted hematopoietic stem and progenitor cells at 500 times gravity for 5 minutes at 4 degrees Celsius. Once the centrifugation is over, discard all but 100 microliters of supernatant without disturbing the cell pellet. If there are less than 5,000 cells and the volume of the sorted sample is less than 0.2 milliliters, first transfer the cells to low-binding 0.2 milliliter PCR tubes before centrifugation. However, if there are less than 5,000 cells but the volume of the sorted sample is more than 0.2 milliliter, then centrifuge the cells at 500 times gravity for 5 minutes at 4 degrees Celsius. After centrifugation, discard all but 200 microliters of supernatant. Then, resuspend the pellet and the remaining 200 microliters